Well, we start with the second talk of this event by the Raising Public Awareness Committee of the European Mathematical Society. Our speaker is Miguel González Velasco from Universidad de Badajoz. Miguel, Extremadura. Yes, yes. You are from Badajoz, but the university is the University of Extremadura. Sorry. Well, Miguel González Velasco is a mathematician expert on the use of branching processes, in particular in genetic and epidemiology. And today he is here with us in order to talk about mathematical modeling of biomedical problems through branching processes. Thank you.
Okay, first of all, I also would like to thank the organizers for inviting me to give this talk. And of course, I'm going to talk about branching processes in the context of biomedical problems. The contents of the talk are the following. I start with a brief introduction to branching processes. After that, I want to give you an overview about branching processes in this medical or biomedical context. And I finally want to focus my attention on how branching processes are useful tools for model epidemics. For modeling epidemics. Okay, in the context of stochastics models, branching processes theory provides appropriate mathematical models for the description of probabilistic evolution of systems whose components, in general cells, particles, individuals, after a certain period of life, reproduce and die. Therefore, we can apply these models in several fields, like, of course, biology, demography, ecology, epidemiology, genetics, algorithms, also the economy. And the former model is linked to the names of Galton, Francis Galton, Henry Watson, who was a mathematician from the University of Cambridge, and also to these Galton and Watson were British, and also the French mathematician Bien-Aimé, and they studied the problem was that the aristocratic surnames become extinct, and they want to solve this problem, or to build a model that can help to understand why this occurs. This is Galton, Watson, and Bien-Aimé. And this model is very easy. I would like to show here how to evolve. We start the process, for example, with one individual, and this individual generates with a certain probability. For example, in this case, with probability P2, two new individuals. This individual from the first generation, and each one of these individuals independently, one of the other, generates new individuals. For example, this one with probability, this one with probability P3 generates 3 individuals, and the other one with probability, for example, P4 generates these 4 new individuals. So that in the second generation we have 7 new individuals, and this generates, of course, more individuals in the next generation. We have 10 in the third generation, and so on. Then the number of individuals in the n+1 generation is given by the number of offspring of each individual in the previous generation, in the nth generation. Then here, x and k denotes the number of offspring generated by the k individual in the nth generation. And Of course, this process, the evolution of the number of individuals in a population, is governed by the offspring distribution, that is the probability distribution that indicate us the number of progenitors that one individual can generate. And the threshold parameter for these models are the average number of offspring per progenitor, that is the offspring mean. Also the variance play an important role. This model, this process is known as BNMA, Galton-Watson branching process, and the evolution of this process has the verified dichotomy, the extinction explosion. That is the process become extinct in the long term or explode to infinite. Mathematically, this is the equality. And of course, the first problem which was studied was the extinction problem. And it's easy to obtain that if the threshold parameter, the offspring mean, is less or equal than 1, then the process died out with probability 1. And if this threshold parameter is greater than 1, then there is a positive probability of non-extinction. And then there is a positive probability that the process explodes to infinity. Okay, this is some sample examples related to the cell division. We have the splitting probability here of a cell is P, and then the cell can split in two or die. Then the offspring distribution is given only by these two values, P2 and P0. The offspring mean is 2 times P, and in this case we can calculate the offspring, the extinction probability. This will be equal to 1 if M is less or equal than 1, and that is P is less or equal than 0.5, or is equal exactly to this fraction if we are in the case in which M is greater than 1. Of course, the other important question related with these processes is the asymptotic behavior, the limiting behavior of the process. And we have introduced here some mathematics. I avoid to introduce mathematical formulas, but you know it's difficult to talk about mathematics without mathematics, and then I decided to introduce these results here. And the behavior is easy to explain. If the threshold parameters, if the offspring mean is less than 1, then this case is named is called a subcritical case, and then the process becomes extinct, and we go to the extinction very fast, exponentially, with the rate given by M, by the offspring mean. If M is equal to 1, this is the critical case, the process will become extinct also in this case, but in this case they take a long time, or it's possible that they take a long time for the extinction, and even in a large generation we have individuals in the population, the distribution of the number of individuals in this generation over the generation follows an exponential distribution. And in the last case, when M is greater than 1, this is called the supercritical case. We have that if the process explodes to infinity, then growth exponentially with the rate of growth given by the offspring mean. Of course, this former model has been modified in several directions in order to fit different real problems. And in this sense, we have several discrete time new models, and of course also continuous time new models. For example, we have the bisexual branching process. This, of course, this bisexual is not related to the sexual orientation of the individual, but only to indicate that there are two types of individuals in a population, females and males. And in this type of models, we consider that in order to produce offspring, we need to form couples, the males and females form couples. And then the process have two phases. The one is the mating phases, so the phase in which the couples are formed, and the second phase is the reproduction phase. Also, we have branching processes with immigration, in which it's allowed that population, individuals coming from an outer source come to the population. Control branching processes, in which the total number of progenitors at each generation are controlled by a random function, or branching processes in varying or random environments, in fact related with the previous talks, with the offspring distribution can change depending on the environment, randomly or in a fixed way, or multi-type branching processes. We have several types, different types of individuals in the population. And also, the continuous time models are, for example, Markov branching processes in continuous time. For these models, so Berman-Harris or Sebastianov branching processes, we have that each individual now have a random variable that control the life length of the individual. And for these first three models, the offspring are produced at the end of the life of the individual. And then we have a random variable that control the life length of each one of the individuals, and offspring distribution that control the number of offspring that each individual have. In the case of Markov branching processes, the left line, sorry, the length life random variable follows exponential distribution. In the Bellman-Harris branching processes is whatever distribution you want. In both cases, in both models, there are the offspring distribution and the left line length life distribution are independent. In Sebastianov branching process, we have a dependency between these two distributions. And finally, we have the Krummel-Jaggers branching process, or the general branching process. In this case, we have that, of course, each individual have a life length, a random variable, a life length, given by random variables. And now the offspring are produced not at the end of the life of the individual, but during this life. And this is governed by a point process. Of course, there are many monographs devoted to the study of these models, from the former to Harris to the last ones. And between these, we can find several of them that are devoted to introduce several biomedical problems, or to solve or to apply to several biomedical problems. I would like to show you here that Kimmel and Asserol, this Co-Jaggers and Battuti. We have also some chapters or small books devoted to special problems, like this branching process model of cancer, or this devoted to the epidemic modeling. of course, I would like to give you very fast, very short overview about the how branching processes are useful tools in this biomedical context. I'm going to follow the scheme given in the book of Kimmel and Asserl. And for example, these models, these branching processes have been applied successfully to solve problems related to genomes or to changes in DNA and chromosomes, in problems related to mutation, coding sequences of DNA, repeated sequences of DNA, gene amplification, recombinations, problems related with cells, cell kinetics, cell division, cell growth, of course, stem cells, cell cycle kinetics, related with cancer, are useful. The last book of Darrett are related with the multi-stage theory of cancer. He introduced multi-type branching processes in order to model this theory, in order to model the different mutations needed that the cancer appear. Cancer cell population, cell cycles resistant to chemotherapy, mutations in cancer cells. Of course, also in population genetics and evolution related to problems, the human immunodeficiency virus, the AIDS, studying the genealogies of the mutant resistant to immunotherapy or to drugs. And also the extension of the real extension of the problem of Galton, Watson, and Bien-Aimé in relation with the surnames, that is the human Y chromosome DNA haplogroups. The study of this problem, or this is linked to X chromosome genes. Our group has introduced several models in these two cases. Several two-sex branching models in order to study the evolution of the number of carriers of alleles, of two alleles, in fact, linked to Y chromosome genes. And we studied the extinction of these alleles, the possibility of fixation of one of the alleles, the condition that guarantee the coexistence of both alleles, and also the rate of growth of each one inside the population. And we have also introduced some two-sex branching models to study the evolution of the number of carriers of course, alleles linked to X chromosome genes directly related with some diseases. And finally, branching processes are also useful models for epidemic modeling. I want to focus my attention now in this part. Of course, mathematical modeling is critical for our understanding of how infectious diseases spread at an individual, at a population level. The branching process has been applied widely to model the spread of infectious disease, at least in its early stages. And the process describing the number of infectious individuals in epidemics may be well approximated by branching processes if the population is close and homogeneously mixing, and the number of infected individuals in relation with the total number of susceptibles are is small. Of course, there are other situations that in which the approximation by using branching processes are also possible. For example, in the cases in which the population is not homogeneously missing, but this population is grouped in small missing units, like household or places. Here I would like to show you how to use the general branching process, the Krum-Jaggers branching processes, in order to model the evolution how evolves disease that follows SAR scheme, that is the individuals can be susceptible, infective, or removed. We have in the population infected individuals, also susceptible individuals, and for example, an outbreak can start with one infected individual. We suppose that each contact produces a new, each contact between an infected individual and susceptible individual produces a new infected individual. Then this infected individual, we are going to the random variable, the process, give us the number of infected individuals in the population at each time. Then this individual this is his life length, that is the period in which he is infectious, and a point process give us the exact time at which this individual is going to meet susceptible individuals and then produce new infections, new infected individuals. For example, in this case, in these three cases. Then, at the end of this infectious period, the infected individual is removed from the population because died or is recovered, but never will be again susceptible. And each one of these individuals produce a new tree. That is, in this case, this is the lifelong period in the branching setting, and this infect two new individuals at this time, and this produce a new branch, etc. Okay, also other models or possibilities of evolving a disease like SEAR scheme, susceptible. Now the infected individual have a incubation period or a latent period, and after that they become infective and finally removed. Several diseases like measles or mumps or avian influenza fit this model, and also the general branching process can be used to model this kind of schemes. Also another well-known like SAS, etc. Of course, in order to control the evolution of the number of infected individuals in the population, the key epidemiological parameter is denoted like R0 is the average number of infections causing a susceptible population by a typical infected individual. In our branching setting is correspond with the offspring with the offspring mean, the mean of the offspring distribution, and then a major overbreadth. We know from our branching theory that some major overbreadth occurs with non-zero probability if and only if this threshold parameter is greater than 1. In order to avoid this major overbreadth, we know that vaccination program represents effective form of control, but usually this is the way in which we work. We suppose, for example, that we have the possibility of a major overbreadth, that is, the threshold parameter is greater than 1, and a fraction alpha of the population is vaccinated in advance of the epidemics. Then the threshold parameter is reduced to 1 minus alpha 0, and then we know that a major overbreadth is almost surely prevented if this new threshold parameter is equal to 1. That is, if alpha is at least this value. This is a well-known result and give us the critical vaccination coverage to prevent a major overbreadth. But many analyses of vaccination strategies in the epidemic modeling literature have focused on reducing this threshold parameter to this critical value equal to 1. However, if the population is large, both in this case, remember the critical case in the branching setting, the total size and the duration of the ovary may be still appreciable. In many situations, it may be desired that to control the duration of an outbreak and the size of the outbreak. And in practice, there is also a cost associated with this outbreak, and to reduce the expected value of this total cost is an important question. Clearly, at least for our population, both of these aims necessitate that R0 is reduced to somewhat less than 1, not only to 1. Okay, I'm going to skip this part. It's also important that we were also interested in how to introduce the vaccination process in population. It's not only to reduce the threshold parameter to a value less, strictly less than 1, but also how to introduce the vaccination process, because this process, we understand, is time dependent. But I'm going to skip this part. And we study these kind of problems, how to introduce this vaccination, how to reduce the threshold parameter to a value less than 1 in this two papers. And we try to obtain optimal vaccination strategies based on the duration of the outbreak and on the total number of infected individuals. We study in this paper these problems, and as an illustration, we analyze the data of mumps outbreaks in Bulgaria from 2005 to 2008. And in this period here, we represent the total number of new infected individuals weekly reported. And in this period, there appears many new cases of mumps. This is the main province in which appear these cases, of course, in Bulgaria. And this may be a result of a pure immunization of these birth cohorts. And of course, provides a supplementary doses of the corresponding vaccine is important in order to shorten the duration of the outbreak, so that our objective was to determine optimal vaccination levels that guarantee with a high probability that the outbreak duration will be less than some suitable bound. We first model the man's outbreaks using the general branching process. In this case, it's necessary to take into account that the young people, that the people that can have this disease are young people, and these are groups in school or universities, and in small separate, and then are a small separate population, and we have a population-dependent propagation, and then the approximation of using the general branching process is valid only when the outbreak are very short. We studied this time. This is the direction of the outbreak that we observed previously. And for example, we have that 97% of the outbreaks have a duration less than 6 weeks. Using this data, we fit our model, and using this model, we obtain optimal vaccination strategies that, for example, give a result like this. Vaccinating 60% of susceptible individuals in the target cohort, we will guarantee that at least 90% of the outbreak will have only primary and first generation cases after vaccination, or if we want with this 90% probability that outbreaks do not spread after vaccination, we have to vaccinate 94% of the susceptible population. Show that branching processes in this case is within a useful tool in order to be used in modeling epidemics evolution. Here is the references that I have used. These are references linked, associated to the problem of Y-linked two-cis branching processes and X-linked two-cis branching processes, and those corresponding to epidemic modeling. I would like to finish with this sentence. Mathematics provides wonderful tools for getting a better life. Thank you, merci beaucoup, grazie mille, and muchas gracias, and all the possible permutations. And my sponsors, and this final picture that I took the other day from the balcony of my house. And yesterday was the day of P. I try to remember. You can find here in the sun cycle the P number. Okay, thank you very much.
Well, questions from the audience? Well, no question from the audience, but as you probably know, I work in epidemics. I use Markov chains. When we use Markov chains, we are able to study probabilistic descriptors, but associated with individuals. For example, the number of infections suffered by a typical individual during an outbreak. And I wonder if, by using branching processes, you are able or not to study this kind of descriptors. I'm not talking about descriptors associated with the population, because it is seen in your talk that you are able to analyze number of infected individuals, the length of an outbreak.
The number of infected individuals, the number of infected, the total number of infected individuals in the populations. But which kind of characteristics you asked me about?
For example, the probability that an individual, a concrete individual, is affected by a disease, for example, during an outbreak. Because, well, you have a population of individuals, but individuals are missing in the population.
Yes, of course. We don't follow each individual separately. We work with the whole population at each time. Of course, in this setting, it's not possible to follow. I think it's to follow each individual separately. You can determine which is the probability of, for example, the probability distribution of the number of infected individuals at each time. This is possible.
Global descriptors.
Yes, this is possible. And from this distribution, you can infer which is the probability that one of them can get the disease.
Okay. Well, thank you. Well, question? Well, in such a case, let's thank again to Miguel González Velasco.